Yeah, thanks for joining us for this conversation today. Now, I gave a little bit of a bio here for us, but before we get started, I'd love to hear a little bit more about your background. Could you tell us maybe what you've been doing the last couple of years and why this conversation around Bowie-Dick is important to you?
You want me to go first?
Yeah, go ahead. Yeah, take it away.
Okay. Last couple of years I've been working for Solventum, which is formerly 3M Healthcare. But prior to that, if you guys were on Monday, you probably heard I worked in sterile processing for about 13 years, and I'm interested in Bowie-Dicks because in those 13 years, a good 7 or 8 of those were overnights. I did the overnight shift, so Bowie-Dicks are near and dear to my heart. I
Mm.
know them very well. That was my life for 8 years.
Yep.
So, and they're one of those tests that I understand that, when you do it so much, you don't really think about it. And so when you start learning about it, it's actually very interesting. So I like to spread that wealth, and I like you guys to get interested in that too. So you kind of understand that, why you're doing it, why it's important. And it's not just one of those things that I got to get it done so I can get going. You want to understand it. So that's why they're near and dear to me. That's what I've been
Sweet.
doing. Tula?
So thank you, Kayla. As my colleague Kayla, we are colleagues. So I work for Solventum for the last couple of years, but my background is chemistry. So I'm a chemist by education and also by practice as well. I've done lots of, I would say a common job for chemists, QA, QC. I've been in QA, QC, and then in different roles in different industries as well. So in polymer paint industries as well, and then doing some research. At some point, I was in university. I was a lecturer. I had my own lab doing some research on different topics, but they all, led my career to Solventum, previously known as 3M Healthcare, and but since the first day I've joined the company, I've been involved and heavily with the just medical device reprocessing, uh, both on the product side, but also standard side as well. So I'm also heavily involved in the standards. Some of you may know me from AMI standards, CSA standards, ISO standards, globally, everything related to standards. So I'm involved with that as well. And definitely there are so many interesting things about this. Bowie-Dick test that has been, it hasn't been basically changed since its introduction, maybe in 1963 till now. So I'm excited about that there are some innovation happening after so many years. So, yeah.
Yeah. Well, I've got some questions around that as well. I'm sure our audience does too. So I'm excited to dive into this topic. It sounds like we've got the right couple of folks in the room to answer our questions today. So I appreciate those. Appreciate those introductions. We'll go ahead and dive right in. And again, just one more reminder to our audience as we're working through this live. If you've got some questions that you'd like to know about how Bowie-Dicks work or some clarification on an answer, feel free to type that in the Ask a Question tool, and I will try to get that over to our presenters today as well. So as we are getting rolling here, could you guys maybe just give us an introduction to Bowie-Dick tests in general, probably the most peculiarly named test in all of sterile processing? What is that pack of cards that we're putting in the sterilizer, and what is it accomplishing for us?
Sure. I can answer that. So, again, if you guys saw it Monday, but I'll just refresh. So the original Bowie-Dick test was actually a stack of towels. So for those of you that may have had to make your own challenge pack, your own test pack, it was very similar to that. It had towels, and in the middle there was a sheet that they actually put. At the time, it was the 3M 1222 chemical indicator, or the type 1 tape on there. And so basically, in that challenge pack, what that challenge pack was testing is it was testing the ability of the sterilizer to remove air from the cycle. And so why that's important is because when the air is removed from the cycle, that means that steam can come in and penetrate every single surface in that chamber. And so why that's important is because you want steam to penetrate every surface, which is like all your instruments, right? You want every single instrument in there to be touched by steam. That's how we sterilize. So they came up with this test in 1963. Where the name comes from is it's Dr. Bowie and Mr. Dick. So that's where we get the Bowie-Dick test. So these two guys came up with it, and published the information in 1963, and we have been working off that information ever since with our Bowie-Dick tests. So your current paper test pack, if you guys are familiar like with your BI test packs, it's the same concept where it's equivalent to that towel pack. And the reason why we have the current ones, which are the pre-assembled ones, is, you know, manufacturers come out with that, and use of that helps with mitigating, you know, variation in making those towel packs. So that helps with standardization, which you guys are familiar. Standardization helps things roll really easy. So that's the newest innovation. And then of course the other innovation we talked about the other day, which is the electronic one. But that's pretty much the history of it.
Yeah, really interesting. I remember several years ago reading the obituary of the late Dr. Bowie, and really fascinating man. Just the way that that the test came to be, and discovering, you know, that the dangers of air pockets in our pre-vacuum steam sterilizers, but then also what he did with his life afterwards. I think the end of his life, he was in Zimbabwe doing different types of care work there as well. So anyway, that's just a teaser for those out there who are curious, who want to go down the hole. Really interesting man to study, not only kind of the beginnings of the Bowie-Dick test, but also the you know, just the man behind it, and his very interesting life that he lived. Well, thank you for that introduction to Bowie-Dick tests. I know that that you said that the key mechanism there is trying to detect if air is left in the chamber. And one of the things that I always wondered about as a sterile processing technician is, how does this little paper card detect whether or not there's air in the chamber? Was there some kind of air sensitivity on the paper that's going on? And I've, you know, my understanding of that has grown over the years, but could you maybe answer the question, like, how does that mechanism work? Like, how does it detect whether or not there's air in the chamber with just some chemical stripes on a piece of paper?
Yep. So, I mean, a very good question, right? It basically adding at what Kayla mentioned, right? The current test packs, we have this sheet, or they can be stripes that they are in center of the pack, right? That they can basically say they detect the air pockets or steam. I mean, existence of air within the steam in the chamber. But the reaction is basically a chemical reaction. So they are also, you know, the Bowie-Dick test, they are type 2 of the chemical indicators. The name is there. They are chemical tests. There's a chemical reaction that will happen. And this reaction, that if it happens completely to the end, that's where we get the positive results, or the result that we want to see. If the reaction doesn't complete for any reason, or it's incomplete or inconsistent, that's a kind of showing a failure. So how does it work is that basically these chemicals, they are sensitive. They're sensitive to heat and the moisture. And when there is air or air pockets, it blocks. When it blocks them, it means that they cannot make the reaction complete, or it will not complete at all surfaces. And as just Kayla clearly mentioned, what is the aim? The aim of this sterilization is that you want the steam to come in contact with all the instruments that you have inside the chamber from all of the surrounding it, not just top part, not the right part, not the bottom, all surface, right?
Right.
So if there are air pockets, what happens? The air pockets are barriers to air. So these barrier to airs will be locating at some parts of, if they exist, they will be locating on some part of those chemicals that they are sensitive to heat and moisture, will block them. And when you have something that's blocking it, the reaction will not be complete. And that's basically how they, if you want to simplify, that's how it's happening. It's air pockets are hindering, are causing barrier for this reaction to be complete, to show this is a complete, uniform, homogeneous color change, which as with the current test packs, basically.
Okay, so the way that it's measuring for air removal, if I'm summarizing what you're saying well, is not by necessarily trying to measure the presence of air, but trying to measure if there's any air impeding its ability to get steam contact. It's really measuring steam contact, and then whenever that fails, it indicates that maybe that there was some air that got in the way. Is that what you're saying? Kind of that buffer in between the two?
So I mean, I was saying equivalent between the two because it takes the air. If there's no air, so then if there's no air means that the steam will be everywhere, so it will be in contact. So the air
Right.
pockets are causing it because we know
Right.
how they design, right? They are working for the vacuum sterilizer, pre-vacuum sterilizer. It means that you vacuumed, so the only thing that caused air will be non-condensable gases and air that will form air pockets that will detect that. So
Okay.
that's a series of things basically happening there.
Okay. I think I've got my mind wrapped around that. So, um, the reason that I wanted to talk about that mechanism is one question that has always haunted me and still does to this day, is what then is the difference between that type 2 Bowie-Dick test and maybe a more robust type 5 integrator test? Because, and just kind of line of reasoning here, like our type 5 integrators require steam contact as well as a number of other parameters in order to pass. And so, wouldn't it also, in a way, be measuring like if there were air pockets in the chamber, wouldn't it also cause a type 5 indicator to fail if the air pocket was keeping the steam to get to the integrator? So I guess my question is, what is it that a Bowie-Dick can do that a type 5 integrator can't do? Why do we have type 2s and type 5s instead of just one test that does it all?
No, it's perfect question. It's
Yeah.
actually just when you get introduced even to this, the whole, sterilization and also monitoring of that. That maybe in early days, that was my question as well. Why this, right?
Yeah.
One, so, and maybe I answered in different stages. So, can a chemical indicator also detect if there's air pocket? If you answer that, but yes, it may or it may not. Depends on that, right?
Mm,
How these tests, we have to again consider a couple of the items. One, when they are being tested, the condition they are being tested, and how they have been designed to react, and what they are being designed to react for. So when we get all these together, basically we'll get the answer. But yes, it may. So an integrator also, it may respond to that. That's of course it's because we have a chemical indicator that's inside the tap. But Bowie-Dick test has been designed for only for basically for the checking if the air removal is complete, if non-condensable gases they are not in the basically in the chamber, and being tested on an empty chamber. So when it's otherwise empty chamber, it's exactly the wording from standards. Because if there is, it might sit, the air pocket might form. Air pockets will not be around. If you have a packet, it will sit on the pack. That's why the test pack is there. And what's its empty chamber, right? Otherwise empty chamber. Because if there is, we want it to gather gear, you can detect it. If you are doing
gotcha.
testing with the integral integrators, again, what they are designed for different purposes. They are designed to make sure that the parameters are met inside each pack, each tray, each container. So when you have a full load, that's when we are using the chemical integrators, then okay, air pocket that we talked about might be on that pack, might not be on that pack, might be on
Mm,
which part of it. So it might be or might not. So that's basically the yes part of it. That can it may respond to that. But again, now you have to focus on the second piece that I mentioned. That when we design them for how they have been engineered and designed, they have been engineered and designed to work in, of course, completely different setting, empty chamber versus the full load. That's the other one. Chemical integrators, they are designed to, as you mentioned, right, to all the parameters that we have, the time, the temperature, and the steam quality. But Bowie-Dick is specifically designed as that just it's not talking about the time. It doesn't. It just not the temperature. It talks. I mean, indirectly, it talks about the heat and the, of course, the existence of the air if it's not complete. So. These are basically pieces of puzzle we want to put together like a quad system. You have to do one check, second check, other check. These are all together. And all the reason going back here, sterility cannot be tested practically. If you want to test something, if it's sterile or not, basically you have to touch it and then it's not sterile anymore. How
right.
we do that, these are all the tests that we put in different stages. One, if we check a sterilizer to work or no, we check the printout. So that's one
Hmm.
check. we check the Bowie-Dick test to make sure that, okay, can it do the can it do the is the air vacuum, is the vacuum complete, is there's no air left, is there no air pockets left, no condensable
Right.
gas left. So we check that. Another, then we do leak testing. Okay, does the sterilizer keep it? So that's another one, we talk about that. So then, and then we have integral indicators. They go inside each pack that we want to make sure, okay, we had this team, did it reach inside every single item or pack that we have in the sterilizer? So that's where they're coming in to make sure that how they've been designed, to that they have passed through the barriers, the packaging and everything, and reached inside each pack. That's what integrated is testing. That's not Bowie-Dick cannot do it, and vice versa. Bowie-Dick can completely check about non-condensable gases, which integrator will not be able to check about non-condensable gases. So that's how they have been designed for. And said all of them putting together, that's what make them, they are different tests for different purposes, but they are all type of chemical indicators.
Sure. All right. Well, thanks for walking through kind of the overlap and differences. One of the things that you said in there that I just want to touch on one more time that I hadn't really thought about is how the presence of trays or packs in the sterilizer might affect where the air pockets are in the chamber, and that's probably why the Bowie-Dick test gets done in an empty chamber. I'm curious, in an empty chamber, if you've got a towel pack that's, you know, this size of a cube, or maybe just a pack of cards that's, you know, this size, that's in an otherwise empty chamber, what keeps the air pocket that might be present in that sterilizer from happening somewhere else in the chamber and not right there on the test card where we're measuring? Like, what keeps us from having a pass at the pack when in reality over in this corner of the chamber there's an air pocket?
So, as I mentioned, if you have so the air pockets that they might be, it's in, and also we have done, you know, testing. These are all coming by experience and the testing. If you have a
Sure.
pack or a tray or something inside the pack, air pockets or what, it will basically will go towards there. And if you have the denser the packet, it will be goes towards that one. When
Right.
you have empty chamber, it's just, you don't have it might be somewhere we don't know, right? But when you have this pack located, it's going to be located there. And where the pack is going to be located, right? What is the most challenging part? It's going to near the coldest spot. It's going to
Hmm.
basically above the drain part, where the
Okay.
packs, where the test packs being tested. They said that what is the most we always say this question, right? We get this question: where is the most challenging part inside the chamber? Where is the kind of card is the coldest spot? Where is the kind of, it could be more, or it could be the more challenging? Ask the sterilizer manufacturer, but in general, we say about the drain. So that's why they kind of is getting close to that part. And when you put the pack there, it's going to be there. But when it's full load, that's quite a different story. Now, when you have a full load, everything is changed. It could be everywhere. It could be on everywhere around that we don't know, might be. But that's the reason that it should be tested on the otherwise empty chamber. And actually, there was a conference recently, WFHS conference, the most recent one. They've done studies about the design. Even the shape of the chamber will change everything. Will change of the flow of everything. Will change of
Hmm.
even the one. So, but with the current systems that we have, with the current sterilizer that we have, said the best way of testing it is just to be otherwise empty chamber for the Bowie-Dick. That's where the air and the air pockets will find their way there.
Yeah, no, that makes sense, and I think I should have thought about that question more before asking it, because it's the point of greatest challenge. It's easy to remove air from the empty corner of a sterilizer, but sucking it out of the center of a tall stack of towels is hard. So if it looks good in the center of the towels and that was the only thing in the chamber, then the whole chamber is good. So I appreciate that. All right, so here's my apology to the audience. I'm a bit of a nerd about these kinds of things, and so let's turn the corner to some more practical questions about, you know, our daily use of Bowie-Dicks and a little bit off of kind of the science and mechanisms for how they work. So first practical question that we want to know, like we know our standards instruct us to do these tests daily. What do you do in the event that a Bowie-Dick test fails? Is that an immediate take the sterilizer out of service? What does the follow-up look like for a failed Bowie-Dick test?
So according to AMI ST79, you know, you should be following your facility's policy. That's the first thing
Okay.
you should do. So just a reminder for everybody, in your sterile processing department, there's policies and procedures for basically almost everything that you do. So Bowie-Dicks aren't any different from that. So your policy procedure may instruct you to take the sterilizer out of service immediately. It may instruct you to run another test just to make sure. Or it may tell you just go straight to your supervisor and let them handle it. But the main thing that you need to do is just consult that facility policy. Um, I know not everybody knows, I used to be that person that I worked part-time at another facility and I was overnights and I was only on the weekends. So I wasn't always sure what to do. The thing is, if you're not sure what to do when you get a failure, make sure that you go to your, coordinator or your supervisor, whoever is in charge on your shift, go to them and consult them and they should know what to do if you get a failure.
Gotcha.
So in worst case scenario, they're not around, you're by yourself, you don't know what to do, just take the sterilizer out of service and make sure you contact either like biomed or, you should have the number to your supervisor or whoever. So
yeah, that's
always
often
consult your facility policy.
Yeah, that's often the safe choice, though. If in doubt, throw it out, kind of thing.
Exactly. Yep.
But yeah, consulting that policy makes sense. Now, there is another test. Kayla, you mentioned being kind of the on night shift for many years. I worked second shift, and so we were familiar with these tests that third shift ran, and every now and then had to stay over and help run tests, but it wasn't our bread and butter. There was another test that was done weekly on our steam sterilizers called a leak test. We ran them once a week on the sterilizers that we had per the manufacturer guidelines, and they always passed on the department that I was in. And I always wondered, what if the leak test were to fail? Is that a different set of kind of triage instructions? And what's it sounds like the Bowie-Dick test is also testing for air leaks. And so what are the difference between these two tests, and what do you do if your leak test fails?
Atila? Well, I'll handle the first part of the question. So if your leak test fails, same concept, there should be a facility policy about that. Just make sure you're consulting that, or
Okay.
if you got questions, always reach out to your supervisor. Now the difference, I'm going to let Atila tell you guys about the difference because he touched on that earlier and he's kind of my leak test guy, so I'll let him explain that for you.
Okay. Atila, I think we've lost your mic. Are you able to hear us? I'm not able to hear you.
Okay, sorry.
Oh, yeah, you're back. Yeah, you're back. All right, go ahead.
Wonderful. Okay, thank you.
Yeah.
So I want to say one thing I always repeat, and I mean also the one that Kayla mentioned, touching that, always follow your what are your procedures, right? What are your internal procedures you have? It's very important. As a person who is very heavily involved with the standards, always, and I'm also wanting that follow your local laws and regulation because they override everything, and it comes down to your local facilities procedures, right? That was when I highlighted that. But getting back to the second piece, what about the leak test and the Bowie-Dick test? They are two different tests with two different purposes that both are necessary. For leak test, even the documents, they mention that, for leak test, again, your facilities procedures, but they say that you follow the recommendation from the sterilizer manufacturer.
Yep.
So let's look at what is leak test. The leak test, and again, they're basically the correlation of that. What's the relation of that between Bowie-Dick test? Because maybe in general, as you mentioned, right, they seem that both they test for, okay, is there air leak? Yes, but in different concept and in different way that they have been designed to. The leak test checks the integrity of the sterilizer, the chamber. The chamber seals are working, the gaskets, the valves, the piping, and even other components. So basically, it won't say that when the sterilizer is in the vacuum, can it hold the vacuum for a certain amount of time, or it's going to lose that vacuum and air will leak through the issue from these gasket valves, piping, or one of these things will happen, and then it will leak into. So what will happen if there's an air leak? The pressure inside the chamber is going to rise. It means a failed um, leak test. That, as Kayla mentioned, what you need to do, you need to inform the personnel and follow the procedures. So it's basically the leak test is done to make sure that it can hold the vacuum. But Bowie-Dick test is testing that if there is no air left, no air pockets is left, no condensable gas is being introduced inside the chamber. So that's what is being tested. And again, they are two different tests, two different purposes that they have been tested, but both are crucial to be done. And even their frequency is different. You may do leak testing once, as you mentioned, maybe weekly, but you are doing the Bowie-Dick test daily. It's but it shows that Bowie-Dick test says that if you are basically performing the vacuum, there's no air left. But leak test says, okay, I test that what is now made, I can keep it for another hour, another for 30 minutes, or whatever the process-wise
Gotcha.
is. So that's the performance. Both of them, they complete each other, but they are not basically competing that one can replace the other. They are two different tests and two different form. And again, their frequency is different, the way of the design is different, and what they test is different.
Yeah, so the leak test that I was running in my department, there wasn't any kind of chemical test put in the chamber. It was something that the machine was doing on its own and then giving a readout. And so our service repair representative, that guy always had some interesting advice for us in the department. And so I always took his opinions with a grain of salt when he was telling me about how things really are, but he described the leak test to me as the machine kind of testing the piping on the back of the sterilizer and the effectiveness of the vacuum pump, of the piping that's going on outside the chamber, and making sure all of that is tight and good. And so you could conceptually have a leak test that was a little bit out of range, but your Bowie-Dick tests are still going to pass because your chamber is well sealed and it's still keeping up with the vacuum. Is that maybe kind of a summary of how it's working, that the machine is kind of testing its own components, but the Bowie-Dick test is testing what's going on inside the chamber?
And basically, that's so. What general question? Have you seen a failed Bowie-Dick test? We have seen it, right? Yes.
I don't know that I have actually, but
okay.
Yeah.
So, but in general, that's why we are testing, right? So,
yeah.
When you see this test, it means that yes, it's possible that you have a basically acceptable leak test, but then your Bowie-Dick failed. So that these are the things happening because it
Sure.
shows that yes, it can hold the vacuum, it can hold what it is introduced in the system. The gaskets, the valves are working, but non-condensable gas gets in, and then your Bowie-Dick will fail. So, but the frequency of that, so how long can it be? That's why it is less frequent, because that's you are checking, as you mentioned, you are checking the chamber, the gasket, and the valves and the piping and everything. So that's why they are checked less frequently than Bowie-Dick testing. And also the way of testing, right? Bowie-Dick test is quick test, you do that, but for them, the technician should be there from the sterilizer. It's not something we can check. Their experts needs to be perform those type of testings, and they are doing, as a less frequent than Bowie-Dick testing. So
Okay. All right. Well, one last question about my favorite service repair rep. It seemed like his favorite test in all the world was the Bowie-Dick test. Anytime that there was something wrong with the sterilizer, afterwards, he would run a Bowie-Dick test just to make sure everything was good before handing it back over to us. If you were coming in on second shift and you were looking through the sterilizer records and you noticed that the cycle count was off for the day, and you go back and look at it and you find this phantom Bowie-Dick test that got ran out of nowhere, Kayla, you know what I'm talking about. It's like, well, the service, it's like, well, the service repair rep was in the department today because there's this Bowie-Dick test in the middle of the day that we can't seem to explain. Why is it that is the test that, at least in my experience, the service repair reps love to run to make sure that the equipment's working right?
So, a very good. I like simplicity, and and again, I'm standard guy. I like something which is standardized and it's simple. So that's where all let's go. You have a simple and effective diagnostic tool. The Bowie-Dick test gives you that. It's a simple tool. It's a simple, quick diagnostic test tool that gives you that. So the simplicity there. It's a diagnostic tool, and then you get the results, and the results are either incomplete, uneven color change, non-uniform color change. You just basically you'll interpret that and you'll get the results. So it is a simple and effective tool. And also it's validated. We are talking about the standard. Bowie-Dick tests are validated vacuum tests. They make sure that air removal is complete, right? They make sure that there is no air left. So if you are doing any vacuum test or leak test after doing tests, if you want to just test it, if you have a Bowie-Dick test immediately there, it's quick test to give you that. And it's a standardized test. We talk about standards, like ISO 11440 series, like 345, for example, part 4. So they talk about Bowie-Dick test sterilizer, how the tests. So how they are consistent or reliable, or what they've been, how they have been designed, that they should be. What are the conditions that you have to design your BD test so that they can work? And then so now you have this pack, the pre-assembled pack. So it is, I said, it's validated, it's standardized, it's quick, and it's effective. So, and you are following basically also standards as well, right? What the AMI standard says, what the ISO standard says. So it's, I would say it's a good tool that they have in their box. Let's do one test and we are good to go.
Yeah, I always just figured it was because it was the shortest cycle that could get them out of there and onto the next facility. But there's probably more to it. I appreciate that backdrop. So that short cycle piece of it, since we're not running these Bowie-Dick tests on just a standard pre-vac exposure cycle, but usually there's a pre-programmed cycle on the machine that we run. And Kayla, I think you covered this in your presentation, but why is it that we have a special cycle? Like, what's unique going on in that Bowie-Dick cycle that really aids that test in detecting air removal, as opposed to maybe a full 4-minute exposure pre-vac cycle?
So, like we mentioned, it's testing. Is the air being removed? Is steam coming into contact with everything that we need? Are there air pockets? Is there non-condensable gases? It's a shorter cycle, too, because we don't need 30 to 40 minutes of dry time.
Well, yeah.
So that's why it's so short. Think about almost like, heaven forbid, you need to run an IUSS cycle, right? Those are much shorter because you've got like 1 minute dry time. Right. So it's the same concept with Bowie-Dicks. You need just enough to get to your preconditioning phase. You get to your exposure time, which is 3.5, maybe 4 minutes if you need to do full minute intervals on your sterilizer. And we don't require dry time. There's no instrumentation in there. So that's why it's so much shorter. That's why you're looking at 23, 25 minutes versus, when you do your load, you're looking at an hour plus.
I gotcha. Okay. So,
yeah. So everything you need is in that short time.
Yeah. No, that makes sense. And that's why we don't want to run instruments in that cycle, because it's not, not going to give you the exposure time, not going to give you the dry time that you need, all of those things. So I want, with you guys live on the air here, to crack open the great Bowie-Dick debate in departments. I think if we were to ask any one of our audience members, like, what's the biggest confusion that you have around Bowie-Dicks, it's probably not about the mechanism or which cycle to push. It's, what do you do after a major repair and you're trying to re-verify or revalidate that sterilizer? Because nobody wants to have to go through the 3 Bowie-Dicks, the 3 biological tests that are recommended in the standards. So could you all help us sort through the debate between which tests come first, the 3 Bowie-Dicks or the 3 BIs, and more particularly why that is? Because I know there's a recommended order, but I don't understand why that recommendation is the way that it is. So could you give us some clarity on that? What's the order and why?
So the order, and I've been through this debate many a time in my tech life. The order is, the official order, according to ST79, is you run your 3 biological indicator test packs, you do your 3 BIs, and then you do your 3 Bowie-Dicks. And the rationale that they get behind this is because, A, this is going to present your greatest challenge, okay, in this order. It's the greatest challenge on the sterilizer. So remember, if you've done a major repair on it, you want to make sure that's working good. You're going to challenge it, right? So this order gives you the greatest challenge, but also it allows time for your BIs to incubate before you do your Bowie-Dick. So while your BIs are incubating, you can run your Bowie-Dicks. And those are tests that, remember, we're taking them right from the sterilizer and we're immediately interpreting them. We're immediately getting the results.
Gotcha.
So that's straight out of SC79.
All right, so I can feel the tide of SPD grumblings growing behind me. So I'm going to play devil's advocate a little bit here and volley some questions back towards you. So I understand that, like, the motive for wanting to start with our most robust test first. That being said, what if I've got, say, a 24-minute readout BI, or one that's even faster? I saw a clip for a 7-second read BI. So I think the market is continuing to move in that direction. Like, if timing wasn't an issue, is there still a clinical argument for doing the biological indicators first as opposed to the BI tests?
Honestly, in my opinion, yes, because it's done in an underwise empty chamber, if you remember, okay? I think a lot of people forget that too. When you run your BIs, please do not run them with instruments when you're doing qualification testing.
That's my next question. So let's circle back to that.
Yeah, no, it's supposed to be in an otherwise empty chamber. Even if you have a shorter readout time, that's still a longer cycle. You know, you want to knock your cycles out that are the longest first. And then do your shortest one. So you still need to allow them to incubate. And it's still your most challenging. You know, you've got that BI in an empty chamber and you're testing the repairs on there to make sure it's working correctly. So I still will go with it. You know, yeah. I still would recommend it. There's a clinical reason why. I think, yeah, we got shorter BIs now, but I would still follow the process.
I gotcha. Okay. Well, I appreciate you sticking to your guns there. One other objection that I've often heard about this, and you can respond to it or choose not to, is, you know, if the machine is broken, it's likely not going to pass the BI or the Bowie-Dick. And my Bowie-Dick test, I get the results faster, and they're a little cheaper for me to buy. And so, like, they would rather know sooner rather than later. So they want to run the quick test first. And if it gets through the Bowie-Dicks, like, then we'll step up and do kind of the next stage of the more expensive, longer test. And then when it gets through those, then we reintroduce instruments. So I can understand at least the motive behind wanting to kind of stage into it so that you can discover, you know, quicker, sooner, cheaper, that the machine isn't working. But I also get the clinical argument that you're making about wanting to start with your most robust test first. So that makes sense. All right. Back to the instruments question. So there was a department that I was talking to that was wanting to validate their sterilizer after a major repair, putting instruments in there in the biological test portion of it, but then they just held the instruments off to the side in quarantine until all the biological tests passed, all the Bowie-Dicks passed, and then if everything passed, then they kept the instruments. Now, I was sympathetic to this department because they only had one sterilizer, and so whenever they had to do this qualification, half a day's work was gone for them because of the amount of time that it took. And so I understood the motive and kind of the reasoning for how they got there. What do you think about that kind of practice, and maybe what should have that department been doing?
Atila, you want to give a little input? Yeah. I think, just me personally, I wouldn't risk putting instrumentation in there. I mean, even if you do it and it's fine, you're still running that risk of it's not. And that's rework that you're still going to have to do. And that's going to take even longer than if you would have just waited and got your results and then you were able to do it. In the case of qualification testing when you're down, I think a good plan on working around understanding that you have qualification testing that's coming and you have to prioritize your instrumentation. I think putting a plan in place for that to help mitigate anything that can happen is a good idea. So often when we had because, you know, I worked at a smaller facility. I worked at children's facilities, and, you know, we had, at the time, we had 2 really large sterilizers, and then we had, if you guys are really old school, you know what I'm talking about, the really small sterilizer with the wheel.
Yep.
Um,
The
and
the
so
submarine door on
the
the front.
submarine door.
Yeah.
And it was a it was still, 2 cubic feet, but it was little. So it was about the
Yeah.
size of a IUS sterilizer. So when we were down one of our regular size ones and we had to do qualification testing, we would have to plan around prioritizing,
Right.
looking at the schedule for the day, looking at, making sure we're watching for add-ons and just prioritizing as the day went to get the instrumentation out. Um, Better than risking putting it in while we're doing the testing and it doesn't go as planned, you
Right.
know. So that's just my opinion from an old tech perspective.
Yeah.
But Attila, I mean, if you want to go ahead and give some input.
Just, I will add just one more point to that. I totally agree. Planning ahead, it's very important because even if it, just in a general one day standard, there are some statements mentioned that lack of inventory or time or others, they should never be a reason for you to not to follow or try to do some shortcuts or steps. So I understand all the, as you know, I definitely understand the pinpoints there that really can be, time wise, resources wise, but none of them can be a reason based on standards to not follow the standards. That's what basically is mentioned and highlighted. And one thing can help is definitely planning ahead. Planning ahead can help us at least reduce the amount of those shortcomings for us.
Sure. Okay. Well, I appreciate that, and I think planning ahead, having a contingency plan, obviously having some redundancy in your sterilizers is helpful. But if not, figuring out, are there any other small chamber sterilizers that you could program a terminal cycle on to be able to get priority things through while your big one's down? Lots of other things that could be done. We're not ever totally stuck. So, all right. So while we're talking about the order of testing, I also think about the order of daily testing. So many departments that are shutting their sterilizers down overnight are coming in. They're doing a warm-up load if that's what's required by their manufacturer. They're doing a leak test. They're running a Bowie-Dick. They're running a BI test. Does the order of those four things, warm up, leak, Bowie-Dick, BI, does it matter, which sequence they go in, or can it be any as long as they all get done in the day?
Order matters. Very much so. So you got to remember, leak tests, obviously you're going to do it per the recommendation of your sterilizer manufacturer.
Right.
You'll do it at that frequency, right? But your Bowie-Dick test, now I talked about this on Monday, you want to make sure that your chamber's warm. You want it up to temp, because if it's not, that could impact the performance or the results that you may get for your Bowie-Dick. So always make sure your sterilizer's warm. As you mentioned, if you're shutting it off at night and you turn it back on, run that warm-up cycle before you do it, so you make sure that the chamber is up to speed. Even if you're not shutting it off and it's just sitting for a lot of hours, make sure that you're running that warm-up cycle. Now, sometimes 24-hour facilities, you're constantly running that thing, right? So you may be able to omit the warm-up cycle if that's the case, because it's up to temp. You're
Right.
running it. But you want to make sure it's warm. Okay, that's your first thing. And then you want to do your Bowie-Dick before you do your BI, because remember, this is your equipment check. You're checking to make sure that your sterilizer is in proper working function for the day. Okay, so that always needs to come before your BI, because you're testing to make sure my sterilizer is good. I can release it for the day, and then I can do my instruments, which is what you're going to use your BIs to monitor. Okay, so that's why the order is important. I'm warming it up. My sterilizer is good to go. Now I'm going to actually use it for the day.
Sure.
So and I think you do it a little backwards, it's not.
I gotcha. I think that makes sense. And every department that I've been in always had, a very strict order about things. It's just one of the things that I wonder, if you step into a department that flips the order, is there anything wrong with that? So definitely take that word then and keep your order straight. Follow the kind of logical steps of testing. We are primarily talking about pre-vac sterilizers when we talk about Bowie-Dicks. That's where the Bowie-Dick test came from, was testing pre-vac sterilizers. But I'm curious, why is there not a Bowie-Dick test of some variety or some kind of air removal test for gravity sterilizers? Because we still have to get the air out of the chamber to get steam contact, right? So there still is air removal that's happening, even though it's not a vacuumed air removal. So why isn't there some kind of air removal test for gravity sterilization? And should there be?
I think I'll get that question. So, I personally think there should be, okay, but there's not. So I personally think there should be one. But as you mentioned, in gravity, right? In gravity sterilizers, we rely on just basically the natural displacement of air by steam. We know that in vacuum, we know that the pumps are doing, and vacuum we have happening. But in the gravity, we just said, okay, naturally the air will be displaced by steam, and we rely on that. And so a test has not been developed for that one because, again, how it works. And in vacuum, we know that there's a vacuum, so you can test that. But in this one, it happens gradually. So when are you going to test that one? So, and because of these are some of the reasons that they maybe they weren't, but it doesn't mean that they may not have in future. If they are basically, if they found a way how we can test it properly, then it will have some benefits. One, we'll have another quality check also for gravity sterilizers. Then we can have a test, basically, you know, how we are doing it with Bowie-Dick test with the vacuum and pre-vacuum sterilizers. Now we can have the same certainty for the gravity, that we are
Mhm.
sure that there is no basically residual air which can cause problems, especially if there's incomplete air removal. No matter if it's gravity or if it's the pre-vac, it's going to cause problem. And
Right.
especially with the some complex instruments that they may have loads with restricted areas, right? So existence of that will be very beneficial, but because none has been designed or standardized, we are again. I think I mentioned standards a lot. It's my passion. I cannot get away from that.
It
Uh,
shows.
it should be some standardized and validated method, and because of that people should agree on that. And when I say people, means all the stakeholders. It's not just industry and user regulatory side. When all these people, the experts, engineers, they should come and they should agree on that. Okay, this is a standardized method that we can use, and it's not hindering the process, but also can be easier, like a BD test. It's quick test, we can do that, right? If we come up with those at some time in future, why not? They might be one, another standards will be developed for that, and then the products will be out there to be used. But currently we don't have. But I personally think if it exists, it will be beneficial because, as I mentioned, we can have we can ensure that there's a better air displacement. We can have another extra quality control assurance there, and basically a standard sterilization reliability. It will help with that as well.
Yeah. Uh, yeah, well, thanks for running through that. I mean, just in layman's terms, thinking about it, you would think that gravity sterilization would be at even higher risk for potentially having an issue with air removal than a pre-vac sterilizer. And so, something that I've always been curious about. Maybe the future of technology will help us in that
Yep.
regard. So as we're thinking about technology advancing, you know, what has prompted the conversation, both in, you know, earlier this week in the webinar and then our conversation today, is this entrance of electronic Bowie-Dicks entering the U.S. market. So could you give us kind of an introduction, kind of non-product descript, but just at large, what is the functional difference between kind of the traditional Bowie-Dick tests and how these new Bowie-Dicks work? Like, are they measuring the same thing? Is there anything distinctly different about them?
They're measuring the same thing. So obviously, air removal, steam penetration, um, looking for non-condensable gases, all of that. The main difference is the mechanism, how it works. We don't have a test sheet anymore.
Right.
It is now a lumen-based challenge, so it mimics, you know, air removal from lumen instruments, similar to that. So I would say that's more of a robust challenge. And then, you know, the steam will penetrate down after the air is removed to the electrodes, as I talked about on Monday. But
Hmm.
it gives an electrical resistance reading. And so you're getting a more accurate reading on the performance of your sterilizer. So that's the main difference in how it works. The main difference between the two, though, is now we're taking out that interpretation part. Okay, you don't have to do guesswork.
Right.
it'll tell you. And so
Pass, fail, green, red.
fast,
Yeah.
yeah.
Yeah. Yeah.
And you may have been in that position. I've been in that position. Well, maybe not, because you mentioned that you've never seen a
I've
fail
not seen a
before.
failed one. Yeah.
I have. And I've been in a position, where I'm like, is this really a fail? I had to call my friend over, and I'm like, look at this. Does this look like it? And we would have a debate, and it would be,
Yeah.
let's take it out of the department. Let's look at, there's no windows in sterile processing sometimes. Those of you that have windows, you're very lucky. I'm very jealous. So, we would have to go somewhere where there's more light, to make that decision. And so, imagine if you were by yourself and you didn't have anybody to run that by and you had to make that decision. This takes that out of there for you. It gives you a true, accurate reading of what your sterilizer is doing. And it's digital. You have that digital capability. Over the years, we've seen in sterile processing that we've moved from paper everything. And, that's how it was when I started, literally writing every single thing that you're putting on that load, every single result, all of that. Now it's got the capability to just talk to your tracking system that you have. So it's able to make everything quicker. And the one thing that I really like about this, again, being a former tech, it's so easy to use that you can standardize this. And when you get new people in, you don't have to worry about trying to teach them how to interpret that test sheet anymore.
Yeah.
That's something you can just skip over now. It's going to tell you.
Yeah.
So it is the same thing as I talked about. It meets type 2 standards. We did the testing for part 4 and part 5. So it's the same exact type of challenge. It meets the same requirements as your current test pack. It's just now you're getting a more robust challenge and you're getting an accurate reading. And you can trend the performance of your sterilizer over time. It has that capability too. So it's better in a way that you'll know your sterilizer's performance is trending towards a failure before you just get a fail, which can be very detrimental to your department. You know what I mean?
Yeah,
So
because you're getting that historical data. And and the automated documentation piece of it that you mentioned does sound important, right? It takes some of the possibility of documentation errors out of the hands of maybe a learning tech doing documentation, and so that's an important piece of it as well. Atila, you mentioned at the start of our conversation that there hasn't been much innovation in the Bowie-Dick space in decades, and I've heard of these Bowie-Dicks before existing out in Europe because for whatever reason, they tend to always be about 10 years ahead of the US. I'm not sure why that is, but it's just the reality we work with. Why do you think that is that this Bowie-Dick technology hasn't seen an advancement in so many decades, and maybe we're just now getting our first taste of it over here in the US?
What was in basically in Europe also? So some of them are different type testing. Some of them are just basically the data loggers. So again, so they are not Bowie-Dick testing. So data loggers are different system. Again, I don't want to go into details of that. That's maybe outside of our discussion today. But they are different.
Yeah.
But okay, so going back to the question that why not? Sometime might be again. How to say that we get things for granted. Oh, it's working. Okay, so it is working for now. So why not just it's working and let's just make the other one. And then also we are just different manufacturers are making and producing them. And okay, this method's been known. Now because it's known, it's easy to use. It's easy to make. Let's go with that. And why do you bother? Because when it's innovation, you have to invest on it. You have to have engineers. You have the team. You have to do that. But for some companies like us or others that they do innovation, so they always look what are the ways that we can make it simpler or easier for people. So and they invest on that, and that's maybe I believe that maybe that's the R&D piece of it. That not all companies they have these R&D. Some companies are just manufacturers and they just manufacture what they can get, but they don't really invest on R&D piece. And I believe that more companies they should do, because if you have budget for R&D, that's where the innovation technology is coming from. And sometimes we realize, oh, this product is working and it's easier to make. Let's make one, and just like other companies, let's make one and have our own versions. And all different manufacturers, they have their own versions of it. But for really being innovative, you need to spend resources, and resources are of course your engineers, your team, manufacturing site, testing lab, and all other. So they require. And I'm always supportive of innovation, that anything that can make life easier, But of course, sometimes innovation can make it more difficult. So no, not that part. But anything can make life easier, can make it, Kayla mentioned, right? If even just removing some interpretation out might be simple, but it's a lot help. But at least you're not worried about that. Failure pass, simple. You are doing that. If it's helping, it's just one step there. All of these, add up. They add up. If I'm saving a few minutes there, it adds up. These are the things that we do. And it's not just by here. We have a whole process. And it's just looking at one piece of it. We have to look at, when we are doing process improvement, how we can improve our process by doing little pieces like in Kaizen management, and all together it will end up we have a very nice process with well improved process basically. And now this innovation can help with one piece here, one step helping there.
Yeah, well said. Guys, we are about out of time. So I want to have a kind of our closing thought be to kind of dream about the future a little bit. I'm curious from your all's perspective, where do you see the future of air removal testing going? And you can take this any which direction that you want to, talking about gravity sterilization, parametric release, data loggers, Bowie-Dicks, perfect documentation, wherever you want. But in kind of your ideal world for sterile processing, where do you see the future of air removal testing going?
I think I see more innovations as we get, more sterilizers, different, methods and things like that. Instrumentation is always changing. It's becoming more complex. So I imagine, air removal testing will probably be following that even more so, So this is a good leap, because this is a product that hasn't been touched in decades.
Right.
So I think we can only take off from here, and I'm excited to see where it goes. And as we become more digital in sterile processing, I just imagine everything just becoming more, digital focused and less paper focused, and air removal tests following that method. So,
yeah, well said. I'll give you the last word here, Atila. What do you say?
I kind of agree. Sometimes, basically, the first step is that, one innovation, so it might trigger other innovations as well. That might happen. That's what we have seen in industry. So, but I think it's as Kayla clearly mentioned, that in the complexation in instrumentation is not going to be simple. It's going to be more and more. Of course, maybe they become user friendlier to use, but the device is going to be more and more complex. So for that, we need better control, better quality control. So for steam sterilizer that we are pre-vac, I believe it will be more and more innovation. But now, again, future is not just about this, but we have now other modalities. We have now other sterilizer. We have all innovation we can see with all other sterilizing agent and sterilants coming up with different sterilizer. So innovation will happen. That's the way, because I said sometime even a thing that maybe not touched for a while, but at some point we realize that it's time to do it. So that's what happened with Bowie-Dick. And so innovation will continue, because that's will drive the future.
Yeah, well said. I'll put my tinfoil hat on and throw in my future prediction as well. I would love to see a more inclusive test that just do all of it: air removal, steam penetration, all of the things, and maybe even in real time. Rather than having a sequence of all these tests we're doing, there's just one thing that goes in the sterilizer and tells us about what's happening real time in the sterilizer, measures all the parameters, and we have a higher degree of sterility assurance. I don't know what that's going to look like and how expensive that would be, but I would love to see that in the industry maybe someday. Guys, you have answered a lot of my personal questions. There were a couple of questions that have come through as well right at the end that we didn't get to, so we'll make sure to get those emailed over to you. Audience, you've been great. Thank you for spending an hour with us here in the middle of the day to learn about Bowie-Dick testing. It's my hope that you got some valuable takeaways from this presentation, got some of your questions answered. If you didn't, in the speaker bio tool, you'll see some contact information there for our speakers. I would encourage you to connect with them either on LinkedIn or through email and continue this conversation. I want to say thanks again to our sponsor at Solventum for making this hour of education possible. If you are interested in the CE certificate from today's conversation, I would encourage you to stick with us. Right at the conclusion of the webinar here, you'll be automatically redirected. If you'll join me in saying thank you to Kayla and Atila for their efforts today. Feel free to click some reaction emojis in there to send some love their way. Thanks again for your involvement in the presentation today. With that, we'll go ahead and wrap it. We'll encourage you, as we always do here at Beyond Clean,